want you to make sure you review your endocrine stuff so you're all caught up and you're comfortable with that. Kind of review what we did last time on the slides. So you go over the blood slide, make sure you can identify all the blood cells on there as well. And then what we're going to do also there on page 79 in your lab is you're going to do some blood typing. So we got the Eldon cards. It's kind of a semi-crude way of doing your blood typing, but it's pretty accurate most of the time if you do it right and you get a good blood sample. What you want to do is up here in the front on the cart, we have the little pamphlet. Tells you about the blood typing, goes through it. We'll talk about this in lecture as well. But right on the inside, it kind of goes through the procedures with you of exactly what you need to do. Now on these cards, you're going to take them out of the little foil package and just a little flat card. And it's got 4 circles on it. Okay, 4 decent sized circles. And in each one of those circles it has a set of antibodies. Antibodies that are going to be testing or binding to proteins on your red blood cells. So what are the proteins on your red blood cells? What are they called? They're called A protein and B protein.
Oh.
That damn beats right. So when you do a blood typing, you have type A blood or type B blood or type AB blood or the type O blood. Okay, well if you have A proteins on your red blood cells, you have type A blood. Okay, which are in fact they're also called A antigens. If you have the B protein, you have B blood. So if you have type AB, then what proteins do you have? Right, you have both of them, A and B. And then if you're type O, you don't have any of either one of the proteins. So that's your type O. Well, the other one is the Rh antigen. It's the Rh factor. And with that particular protein on your red blood cells, that particular antigen, you either have it or you don't. If you have it, you're positive. If you don't, you're negative. Okay? And so that's where the positive and negatives come in. So therefore, when you do this test to determine what your blood type is, you got all sorts of possibilities. You can be A positive or A negative. You can be B positive or B negative, AB positive or AB negative, and then O positive and O negative. Okay, so those are all the different variations that you can be. And the way that you do that to test it on the Eldon card here is that each one of these wells has the antibodies which will bind to your proteins. And so what you need to do is when you take the card out, you need to suspend the antibodies first. So you're going to put a drop of water into each one of the circles or each one of the wells, and that'll help to suspend the antibodies in the circle or in the well. Then you need to prick yourself, finger preferably.
Somebody's going to have to do it.
So we have the little kind of semi-generic, but they work, little lancets. And now with these, you see the little orange part on top? You need to, don't just yank it out. Okay, you have to twist it a little bit, and then it should just kind of slide out real easy. And that means it's ready to go. You got the little trigger on the side. You don't want to push it up against your finger. Don't just set it there real lightly. You need to push it onto your finger. Okay, so
I'm like all bone. That's
give it
just
some
going to get me.
pressure, push, and then hit the trigger. And then what happens is the needle just kind of shoots out, pokes you in the finger, and then it retracts. So it's a one-time, one-use only. You mess it up, guess what? You're going to have to get another one and do it again. If you don't get enough blood, you'll have to get another one and do it again. Okay, you need 4 good drops, or actually you're going to need 5 good drops of blood. Okay, 4 for the Eldon card, and then you're going to grab a piece of this, it's called talcus paper, just a little paper, and you're going to put a drop of blood on here as well. You don't have to put water on this talcus paper. So you need 4 good drops on the Eldon card, and 1 good drop on the talcus paper. So you take and get your blood, and when you get your blood to start coming out of your finger, then you're going to take these little sticks, get the blood, and mix it in with the water and the antibodies in each well. You need to make sure you use a different stick, a different stir stick for each one of the wells. Don't use the same one for all of them. Use a different one for each well. Okay, why is that important? Okay, good, you guys actually did it. Everybody else is like, huh?
Wow.
Okay. You got to use a different stick for each one because you don't want to mix the antibodies with the other wells because then you get all sorts of funny results. So that's what you do. You take this one, get the blood off your finger, mix it in the antibodies. Get another one, mix it in the second well, and the third well. So you'll have 4 of these. And if you need to use more of these, that's fine. I mean, we got like a million of them in there. So, and then it's not like
If
these
you
are
put a whole drop in
overly
each one, it's
expensive.
too much.
A whole
A drop like you just
Oh,
dropped.
yeah, if it just drips off, that's probably going to be a little too much. And you won't get that much with these lancets anyways.
What
But-
does the paper make?
What's that?
The paper you have to pull out this paper?
Oh, talcos paper?
Yeah.
I'll get to that. So, get some blood onto here, mix it into the antibodies. Now, when you mix it, I want you to mix it around in the whole circle. Okay, don't just mix it right in the middle and that's it. I want you to spread it out in the whole circle. Now, keep it inside of their own well, inside of their circle. So you have to mix within the lines, I guess. Okay, so don't go outside the lines. Let it sit for about a minute. And on some of them you'll see you'll see the clumping real easily. Sometimes maybe not. You'll have to mix it again. After about a minute, then you mix it again, and you'll be able to see if it's going to clump or not. And that's what you're looking for. You're looking for the clustering or clumping together of your red blood cells. If you have the proteins present on your blood, then the antibodies are going to bind. It's going to cause your blood to cluster and clump together, and that means you're positive for that particular protein. So if your A well clumps together, then you're positive for type A. Okay, so you're going to be working on that. Well, what about the extra drop of blood you put on here? Well, in the talcos paper, you're going to put a drop and just let it dry for about a minute or so, and you're going to test your hemoglobin content. Now, again, this is Moreno Valley College, so we use cheap stuff. There are more precise and efficient ways of identifying your hemoglobin content, but we have these little pieces of colored paper. You would take your blood after it dries and try and figure out which one it's closest to. Okay, so you can kind of generalize. It's not the best in the world, but it still gives you an idea of where your hemoglobin is, and you kind of give you an idea if you're anemic or not. If you're right in this range and it says, oh my God, I'm anemic, I'm going to die. Probably not. You're probably a little bit higher than that. Now, if you're down here and your blood's drying orange, then yeah, I would definitely get that checked out because there's problems. Okay, but pretty much everybody's going to be up here in the normal to suggestive range. All right, so that's what you're going to use the talcos paper for to test your hemoglobin content. Okay, so you can work on that.